Now it is my pleasure to introduce Dr. Donna Fennell, Professor and Chair of the Department of Environmental Sciences at Rutgers University, who is our 2025 Theobald Smith Society Waksman Award recipient. So Dr. Fennell is a leading environmental scientist, environmental microbiologist, and engineer whose work has transformed how we understand and remediate contaminated environments. She earned her PhD in civil and environmental engineering from Cornell University and has since built a distinguished academic and research career at Rutgers. Over the past 2 decades, Dr. Fennell's laboratory has tackled some of the most challenging environmental problems, ranging from microbial dechlorination of dioxins and furans to the transformation of complex contaminant mixtures in groundwater, to innovative work on airborne microbial communities as well as bioaerosols. More than $7.8 million in funding from the National Science Foundation, the Department of Defense, the EPA, and private industry has supported her interdisciplinary research. She has published over 100 publications and has served as a mentor to numerous graduate, undergraduate, and postdoctoral researchers, some of whom are here today. She is also a past president of the Theobald Smith Society and a recipient of multiple teaching and research excellence awards. Dr. Fennell is also a tireless advocate for integrating microbiology with environmental engineering to solve real-world problems, whether in sediments, soils, landfills, or even the atmosphere. So please join me again in congratulating Dr. Donna Fennell.
Well, thank you, Jen. I'm so deeply honored to receive this award, especially seeing my friends and colleagues out in the audience. It means a lot to me. And you heard some really wonderful words of advice from our young investigator recipient. And I just want to add to keep in contact with each other. This is your network, and these are your allies and friends, and be sure that you keep that contact up as you go on to the next phase of whatever you're doing. Don't forget to reach out, because those friendships are really, really important. So today I'm going to talk about remediation. Let me see if I can, this is, now for something completely different, I guess. So I put these definitions here. What is a pollutant? We always start with this in the classes I teach. What does it mean to be a pollutant? And I think we've heard today, microorganisms themselves could be considered a pollutant. Specific types of genes that confer resistance could be a pollutant. But most of the time in my work, I'm thinking about chemicals, right? Chemical contaminants. And so, I also use AI. And so I know it's gotten a little more sophisticated since I had ChatGPT make these images for me, but I just love how it envisions the atmosphere and the subsurface. So our planet, we have problems above us in the atmosphere. We're at the bottom of the atmosphere. We're at the top of the surface of our Earth, and we've caused problems everywhere. So in teaching the course I teach with Lee and Max and Lily in the past on bioremediation, I often want to tell the students about the history of when we discovered that groundwater and other environments were contaminated. And I don't know Henry LeGrand, but I ran onto this paper of his from 1965, and I just love the writing in it. And he writes about the saturated zone, which is the groundwater that fills the spaces in between the subsurface particles that are below our feet. That the upper part of the zone of saturation and populated parts of the Earth may be considered a galaxy in which millions of enclaves of contaminated water are scattered in uncontaminated water. And how we as humans, we have caused that, and we also use that water. So as we extract water, we spread that contamination around. And he proposed a term, and I may take a vote. Is it mal-enclave or is it mal-enclave? I don't know, and I don't know him, so I don't know what he intended. But what he did was study the shape of the contaminated zones in the subsurface to try at that time to sort of back out where are the sources of this contamination coming from. And so this is, an image that is hand-drawn from 1965. But if we go and look at, a GIS map of contaminated groundwater in central New Jersey, we can see that it looks much the same. And we see that galaxy of contaminated zones. Those are in magenta, along with zones of pumping, which are more the green and yellow and orange areas. And if we zoom in on where we are right here at Rutgers, at the Cook Douglas campus, we can also see that, those mal-enclaves that are surrounding us. So here we are, the bottom green dot is the Department of Environmental Sciences, and the top green dot is where we are at Trey's Hall. Okay, so I don't know if I can get my pointer over there. Try this. Yes, okay, so here we are. All right, so we've got contaminated groundwater all around us here. In New Brunswick, there are dry cleaning fluids. We refer to the chlorinated solvents. These are things like trichloroethene and tetrachloroethene that are used to remove greases and fats and oils from clothing, metal, and other things that we need to use. Up here's where I bought my car. All right, so there's a gasoline spill around this entity. Behind my building, there's a large chlorinated solvent plume. There's also some arsenic in the ground water here. Over in New Brunswick, there's a site of polychlorinated biphenyls. And we could look all around New Jersey and find these contaminated sites. So why is this such a problem? It's in the ground, it's below where we are. But in the subsurface, compounds like trichloroethylene, we call these dense non-aqueous phase liquids. These are heavier than water. So when they're spilled on the ground, they make their way through the unsaturated zone to the saturated zone. And then they start to dissolve in groundwater. And this groundwater is water that we use for drinking. It's where wells are installed. And so the molecules of the contaminant begin to dissolve in that water. But they also, the pure trichloroethylene finds its way into interparticle spaces in clays. And also, from here on north, we've got fractured rock in the subsurface. So it finds its ways to those fractures. And there, that pure contamination, that pure chemical can move very far and very fast from where it was first spilled. And so the chlorinated solvents were a big issue starting as we began to understand the extent of contamination. And early on in ancient times, back in the 1980s, people thought that the chlorinated ethenes were just recalcitrant. They're not degraded by microorganisms. And so instead, wells were installed and the contaminated water was pumped out and sorbed onto activated carbon or removed by air stripping. These are some big, huge, actually, activated carbon towers that were installed at the American Cyanamids Superfund site over in Bridgewater. They're gone now. They got torn down. And they were installed at great expense to treat groundwater. But they failed. And the reason they failed, what I was told, is that biofouling occurred. That means that microbes colonized the activated carbon particles and clogged everything up. So those organisms were definitely growing on things in that contaminated groundwater. So, you know, starting in the mid-80s, I would say environmental engineers and scientists and microbiologists were working on things like chlorinated solvents, which are, as we saw from the map of New Brunswick, incredibly prevalent groundwater contaminants. And so a couple of different modes of microbial transformation of these compounds were discovered. One, you know, it's really not known so far that there's anything that aerobically directly oxidizes a trichloroethylene molecule. But methanotrophic microorganisms, these are organisms that grow with methane as an electron donor and oxygen as an electron acceptor. They have a methane monooxygenase that accidentally oxidizes and makes an unstable epoxide from trichloroethylene. And then you get abiotic degradation of that epoxide. And so at the time when this was sort of beginning to be realized, I was a young master's student at Cornell, working in the lab of Bill Jewell. And Bill recently passed away. I was very sad to learn that. But he had a huge group of researchers. You can see how serious everyone is, except for me. I'm the only one that's amused to be out taking photos. But Bill had a huge amount of funding at that time, sort of at the end of the 70s into the mid 80s, to investigate anaerobic digestion as a way to make methane and make bioenergy. And I think there's a lesson in this. You know, that was at the end of the Carter administration, where fuel prices were very high, renewable energy was very, you know, being investigated. And then that kind of went away. And at that time, there was an emerging funding surge in remediation. So the same funders who were funding him to make methane now started funding him to use methane to degrade groundwater pollutants using methanotrophic organisms. And so he just seamlessly, you know, changed directions in his research. And I think that's, in these times, a good message for us. You know, how do you reframe? How do you keep going? So here you see some methanotrophic biofilms. They have this beautiful salmon pink color growing on diatomaceous earth and activated carbon particles. And incredibly, in his lab, we ran these rather large-scale reactors where we bubbled oxygen and methane into water and then joined that water together to feed the microbial biofilms. I can't imagine REHS allowing us to do that here, but nothing went wrong. It all worked great. And so this was some of the first science that I was really involved in from the beginning, building reactors, growing microbes. And I learned so much from Bill during my time in his group. Now, a little before the, you know, about the same time, a little before the methanotrophic work on chlorinated solvent degradation was published, people had begun to understand that there were anaerobic microorganisms that could dechlorinate compounds like trichloroethylene and tetrachloroethylene that you see here. Tetrachloroethylene is also called perchloroethylene. That's why you see it as PCE. That's dry cleaning fluid. So if you've ever brought a suit home from the dry cleaners and you like that nice smell, that's PCE. So these organisms are anaerobes. They use these organohalides, organic compounds with halogens, as their electron acceptor. And many of the most important ones use hydrogen gas directly as their electron donor. And they knock off chlorines, and they live and grow And for many of them, that's kind of all they do. So some of the key papers were published by Perry McCarty and his group in the mid-80s. Here you see a paper. They were also making methane in attached biofilm columns, and now they switched to feeding their methanogenic columns TCE and PCE instead when new research areas emerged. Francis Parsons and colleagues at the Drinking Water Research Center in Florida about the same time published information about transformation of the chlorinated ethenes, as you see here. And then my PhD advisor, Jim Gossett, and his student, David Friedman, published a key paper in 1989 showing that not only could the tetra and trichloroethene be dechlorinated, but it could be completely dechlorinated so that the end product was ethene. And this was the first paper that showed evidence of complete dechlorination to ethene. And that's important because all of the upstream chlorinated ethenes are known or suspected carcinogens. They have maximum contaminant levels in drinking water, and so they're not good endpoints in a groundwater aquifer. But ethene, of course, is made by plants. It's non-toxic and not a problem as an endpoint for remediation. So Jim, who had done this work with his students, began working with Steve Zender. Many of you probably know Steve. He recently retired from Cornell, as did Jim. And Steve's lab isolated the first Dihalococoides, and he called it Dihalococoides athenogenes, strain 195. 195 refers to January 95, which was when he thought he first had this organism in pure culture. In 2012, athenogenes was renamed McCartia in honor of Perry McCarty. And I just want to note that both my master's advisor, Bill Jewell, and my doctoral advisor, Jim Gossett, were Perry McCarty's doctoral students. So one person, right, has this tremendous impact in educating future students who educate students, and it's just a beautiful network. So students, keep in touch with your network. Now, since that time, many other organohalide respiring bacteria have been identified, Dihalococoides, along with Dihalogemonas, Dihalobacter, Dihalobium. These are called obligate, and that's because all they do for living is dechlorinate a halogenated compound and use hydrogen as an electron acceptor. So they're very limited physiologically. And there are other types of organisms as well who have broader physiological capabilities. So we began, when I joined Jim's lab, we began working on this issue of how to stimulate these organisms in groundwater aquifers. Because hydrogen gas is also not a gas that you'd want to carry around in a big tanker truck and try to sparge into groundwater. So the way that these organisms probably live in their natural environments is that they live in proximity to a complex community that ferments other organic compounds to hydrogen, and that fuels the dechlorination. And so one of the issues in groundwater remediation is how do you then add that organic matter to get the hydrogen without fueling lots of other things, including methanogenesis, because you don't want to go under a warehouse to treat groundwater and have an enormous plume of methane be evolved under that warehouse that then finds its way inside and causes explosive conditions. So my doctoral work was all about looking at the energetics of the fermentation of different organic compounds like fatty acids like butyrate, propionate, also ethanol and lactate, and looking at when the organisms responsible for their fermentation produce this hydrogen, they're sort of self-limited by thermodynamic limitations. For example, if you look at butyrate, it's producing hydrogen at about 10 to the -4 atmospheres, whereas things like ethanol, lactic acid will produce several orders of magnitude higher hydrogen. And when we can produce low and steady hydrogen in a system, that means that dechlorinators can compete for that hydrogen because they have a lower hydrogen use threshold. They have a higher affinity for hydrogen than methanogens. So there's some competitive exclusion of methanogens under those conditions. So here's Jim Gossett. Here he is when he came to Rutgers. When I first came here, he came to a hazardous waste conference, and here he is with Lily Young. And he and Lily were at Stanford at the same time, so they were old friends. You may have been on his committee, Lily was on Jim's committee. Okay, students, so you see how limited your network is, right? There's no escaping it. Okay, so the other thing we worked on at that time, along with many others, was detecting these organisms in groundwater and in aquifers undergoing treatment. And, we had a study on that. DuPont scientists did a lot of work on that. Frank Loeffler, many others, developing, at the time, what we considered sophisticated molecular methods to detect an organism out of these complex environments. And so this body of work that was done in the very early to mid-90s laid the basis for companies that now sell slow release electron donors to add to groundwater aquifers, sell bioaugmentation cultures that contain D. helicocoides. And some of the very interesting and novel kind of creative things that are done in treating groundwater, one of the constraints is we can't put things into groundwater that are toxic. So the fermentable organic matter that has to go into the groundwater should be things that wouldn't harm us. So you see things like vegetable oil, lactic acid, and so on being used. And now I was talking to Sage and Paul earlier. They had the poster on pitcher plants. I don't know if you guys saw that, but they were feeding their microbes something called cricket powder, which is basically chitin. Now, I have never heard of cricket powder being used in groundwater treatment, but I think there might be an idea there for a very entrepreneurial person who wants to make a new company growing insects for groundwater remediation or something. All right, so after I had been at Rutgers as a postdoc with Max, later on became a professor in environmental science, but with Max, we went to new lows. Well, not too low. We went to sediments. And, here's Max and Lee and I celebrating, I think, getting a CERTIP grant. I think I was told never to show this photo, but anyway, here you go. You guys have it. And so with Max was working with much more interesting compounds than chlorinated ethenes. Those are easy compared to the things Max works with. And so when I came here, I started working on the polychlorinated dibenzo paradioxins and dibenzofurans with him. Here's their structures. The PCDDs, the dioxins, there's 75 different congeners of these. There's 135 different congeners of the PCDFs. 17 of them have what are called the 2, 3, 7, and 8 chlorine substitutions. those are the ones that are extremely toxic to us. Okay, so although there are a huge number of congeners, there's sort of a down select in what is monitored environmentally, because those are the ones that get into fish, that get into our food and cause us problems. So dioxins are never made on purpose. They're accidental byproducts. Well, companies that sell the standards make them, but their presence in the environment is all from natural or human processes where they're just made accidentally through combustion or chemical manufacturing. And I don't think Gerben is here, but Gerben Zylstra, another person I'll talk about later, has done a lot of work on looking at aerobic degradation of dioxins and furans that have very few chlorines. When they have less than, say, 2 chlorines, they can be aerobically degraded. But if they have additional chlorines, they're not amenable to aerobic degradation, and only these organohalide respiring bacteria can transform them by knocking off chlorines. So one of the first things I did when I came to Rutgers and worked with Max is we, of course, brought strain 195, worked with Steve Zender and Ivana Nijenhuis, who was also at Rutgers, who's now in at UFZ in Leipzig, and tested strain 195, which has within its genome, it has something like 15 different reductive dehalogenases. These are the enzymes that the organism makes to remove chlorines and reduce those chlorinated compounds. So we found that strain 195 does reductively dechlorinate a whole variety of aromatic compounds, including some dioxins and furans and PCBs. And then later on, I and my student, Fang Lu, she did a study on hexachlorodibenzofuran, which is another 2,3,7,8 substituted congener, and found that this organism takes off chlorines to essentially make end products that aren't really monitored by regulators. Only the 2,3,7,8 substituted ones are. So the kind of magic of dechlorination of dioxins is that just taking off one chlorine in the right position means that although you still have a chlorinated dioxin there, it's not one that interacts with the physiology of our cells to cause the incredible harm that the others do. So this is with Fang, of course, Max, and Hans Rieschnau. Fang also did some stable isotope, some compound-specific isotope analysis looking at the isotopic fractionation of the dioxins during dechlorination. And Weilin Wang, my other colleague in environmental science, was also a member of her committee. And we did lots of other things with strain 195. Here's a pathway of dechlorination of a pentachlorodioxin. Again, taking off key chlorines means that we can essentially detoxify a sediment and remove that toxicity that's so harmful. So in the meantime, other strains of Dihalococoides have been isolated. CBDB1 was isolated in Germany by Uta Lechner and her group, and studied further by others in terms of its pathway. And one of the things I want to point out is just the differences in the different strains of Dihalocoides. That's because they have different suites of reductive dehalogenases. So they may preferentially take off different chlorines from the same molecule. And what that means is that out in the environment, right, we may have numerous organisms in an environment that are attacking these compounds in such a way to produce different end downstream metabolites. So the next thing that I really started to work on was, okay, what are the organisms that are present in one of our most famous, maybe the most famous dioxin Superfund site in the world? And who knows where that is? Okay, so it's right up the road in Newark. Okay, so we have the Passaic River here. Here it is with its enclaves showing. But in Newark was the site of production of chlorinated herbicides. And the famous one is Agent Orange, which is a mixture of two different herbicides. During the production of those herbicides, dioxin was formed as a byproduct. Here's a reach of the Passaic through Newark. The Superfund site where Agent Orange was made is right in here. I actually had sediments from over on the other side of this that John Reinfelder, his student, came down the hall or the bay to our lab and said, "We've got this jar of Passaic River sediment. It's been in our refrigerator for 4 years. Do you want it?" And this stuff is so incredibly difficult to get that we were jumping up and down in joy. Later on, my students and I have gone to other locations along the bank and collected sediments. But here's sort of an overview of that Agent Orange and why, you know, it's such a problem in Newark, because the factory was built right on the river. Of course, this is a problem in other parts of the world. Max has worked with scientists in Vietnam to look at dioxin dechlorination in soils and sediments there, because this was sprayed extensively during the Vietnam War. And up in Newark, you know, there's a Superfund site. Right now, the EPA plans to dredge the lower 8 miles of the river and cap it with clean capping material. But to date, that hasn't been done. And these things have been in the river for probably close to 80 years. They're very present at fairly low levels compared to other types of contaminants. They're very insoluble and hydrophobic, meaning they stick to the sediments. But because they're so toxic, even the small amount that can accumulate into living organisms that live in proximity to the sediments, that gets bioaccumulated as little fish eat that, big fish eat little fish, we eat the bigger fish, and we get exposed to those compounds. So despite the low level, because things like this 2,3,7,8 tetrachlorodioxin are so toxic that it's very problematic. And, you know, dredging is sort of the treatment of choice right now up in Newark. But we know that those compounds, our rivers in New Jersey are tidal. So the sediment gets sloshed around and pumped around all over the New York, New Jersey harbor. And even if the hottest spots are dredged, these compounds are out in our environment. So we wanted to study, you know, who's present in the Passaic River that can dechlorinate 2,3,7,8. So the overall, you know, sort of approach has been to burn through a bunch of undergraduate, graduate students and postdocs to do this work. And I'm showing some of them there. I'll highlight work of Rachel Dean. She did a lot of the work I'll show. Hyder, he did work on the aerobic side, and I'm not going to show that today. Eric Nitzberg, an undergraduate who worked on it. J.C. Miranda, Cassidy Snyder, these are all undergrads. And current undergrad, Nate Avaging, I don't know if he's here today, but he had a poster. And then my current postdoc, Danielle Delp, grad student Melissa Duval. So a lot of work on some really slow stuff. Anyway, so that Passaic mud that came out of John Reinfelder's refrigerator, one of the first things we did was just feed it some other chlorinated compound like trichloroethylene. And you can see that that 4 years of storage did not bother it one bit. It dechlorinated TCE to ethene within weeks. Dichlorobenzene, we also selected that as sort of an alternative halogenated compound because it has kind of a similar structure to the dioxin. You can see that it rapidly dechlorinated dichlorobenzene to a monochlorobenzene. And we used the original sediments and some of these enrichments to test dechlorination of 2378 and 27 and a model congener. And so here you can see Rachel's work and Cassidy who helped, and Hyder, with this incredibly slow process. And you see here the timeline. Grad students might want to cover your eyes at this point, but this says 880 days. All right, so a small portion of the original 2378 that we added to the sediments because we wanted to add more than is there in the background, is getting dechlorinated from a tetra to a couple of dies. And again, tetra is considered the most toxic of these compounds, but once one chlorine is removed, you know, regulators are not really monitoring that. It also dechlorinated the dichloro, the original sediment did, again, a slow process. And Rachel did a lot of work to try to identify what the organohalide respiring bacteria are in these sediments. And she found that although there are dihalogenamonas, there's a very small amount of dihalococoides in our enrichments. And there was a newer phylotype that is probably a new genus that appeared to be most closely most likely stimulated in the presence of the dichlorobenzene and the tetrachlorodioxin. So we're continuing to study that system. And yeah, now we have enrichments that we can use. Now, how many of you grad students recognize this guy? A bunch of you, okay, all right. So when I came here as a postdoc, Gerben, you know, had a big lab, and he still has a big lab, but we were all terrified of Gerben. And I was a postdoc, and I was scared to death of it, you know. We had to go to his lab because he had the poster printer, and people would warn me, you know, Gerben's gonna give you a hard time about your poster, just count on it. And, you know, there were stories that he would come out of his office and watch someone's poster print out and point out the errors, which for me would have been, yeah, there would have been errors, you know. So, but over time, I learned that Gerben is actually, as you all know, you grad students know, he's the best. He's a wonderful colleague. And now Gerben and I, finally, after all these years, we're working together on a Superfund project with Michigan State. And we have a new system to look at. It's the Tittabawassee and Saginaw rivers in Michigan. And it is contaminated also with chlorinated dioxins and furans, but you can see the profiles of the different congeners. The Tittabawassee has more dibenzofurans, whereas the Passaic is a little bit more of a mixture. So we're going to start looking at tetrachlorodibenzophuran in both the Passaic and the Tittabawassee. And we've got, and this is the work that Danielle is doing now. And so we've got samples along the Tittabawassee and Saginaw rivers that we've got a variety of studies started with. For our spatial series, we wanted to we have a relatively limited amount of the sediments, so we decided to start with just an activity test of dichlorobenzene. And so we've got these little microcosms fed with 1,2-dichlorobenzene, added electron donors and hydrogen sources. And now we've starting to see, and this is work by Danielle and Nate, that in these sediments, we also see relatively rapid dechlorination of dichlorobenzene to benzene. Along this segment, we've got one where the dechlorination hasn't been as fast. But for the most part, we're seeing kind of uniform activity along here. So we're a long way from characterizing this, and we're still a long way from being at a point where chlorinated dioxins have an industry of biotreatment, like for the chlorinated ethenes. There is hope though, because for polychlorinated biphenyls, there's a group at at Maryland, Kevin Sowers, who is commercializing some of the organisms and methods for treating those compounds, which are, you know, pretty related to the dioxins and furans. So we're going to continue to work on this and hopefully make some progress, okay, along with others. All right, so that's the below. That's the below. Now we're going to go above. All right. Okay. So when I came to Rutgers and joined the Department of Environmental Science after being in Max's lab for a couple of years, I met this guy named Getty Manelis. And one of the things Getty does, well, Getty does many things, but he is an expert on particles in air, especially microorganisms in air. How do you collect them? How do you characterize them? How many of them are there? And when I met Getty, I knew that from all our contaminated sites, there's literally a ton of anthropogenic chemicals going into the atmosphere all around us. So I said to Getty, what do the microbes in air do? This was about 2005. And he said, nobody knows. And so I started working with Getty, and we began to work on, you know, what are the microbes in the open air? Are they active? Can they transform anything? It's known that there are microbes in cloud water droplets, and they are active and they are doing things. So when you're outside and you see a big cloud come over, that's like a bioreactor right above your head, you know, right over you. A very, you know, full of microbes doing who knows what. But what about the air? All right, so what are the most urgent chemicals that we need to think about in the atmosphere? And of course, there are lots of solvents and gasoline compounds we could worry about, but the big ones for our planet are, you know, might be methane and carbon dioxide. And of course, they're microbial substrates, and so are microbes transforming those in the air. Here's my dad. I said to my dad, I need to go to a gas well and take a picture. So I come from Arkansas, so my dad put his hat on and took me down the road. And of course, he being my dad, I didn't dare walk right up there, but you know, there he is, probably touched it and who knows what he did. All right, so gas wells are a source of methane to the atmosphere when there are leaks, but there are many entities across the world tracking methane emissions. And here you see one such map looking at sources of natural gas or methane to the air from oil and gas, coal, of course, other things that we do. We make we grow rice, we cultivate cattle, we have landfills, all of these things put methane in the air. There are also some super emitting methane areas. This one's in Turkmenistan. And so to me, as we are looking down from space with a satellite seeing methane, here's another mal enclave right up in the atmosphere over our heads. So here they are again, all around us, above and below. So we began to work on, microbes in the atmosphere. Are they active? What are they doing? And so this team included, here's Lee again, Lee Kirchhoff, Getty Manelis. Here's Getty working on some of our air reactors. So we know how to make microcosms and serum bottles. So we bought 55-gallon drums and made air reactors, air microcosms out of those. And in these reactors, here's Kevin, we can aerosolize microorganisms. They are held in the airborne state, and the reactors turn on this roller, and that imposes a bit of a centrifugal force on the bioaerosol, so that holds them in air for some time. For days, we can hold a particle the size of a microorganism. And so we did work early on with Sphingomonas. It's an organism, it's aerobic, it grows on a variety of carbon sources, but it's aerotolerant. And so it was isolated from air. So we started with that. We did experiments in our reactors where we aerosolized Sphingomonas with and without a growth substrate, and just looked at whether those cells made ribosomes. So we did this with Lee Kirchhoff. And indeed, when the bioaerosols are fed, they have a higher ribosome content, indicative perhaps that they're metabolically active, that they are starting to degrade substrate. And then Kevin and Val Krumans started working on phototrophs and methanotrophs. And so we're back to methane, we're back to methanotrophic organisms. They made cultures from the leaf environment and just from outdoor air, fed those methane, and along with Lee, we did stable isotope probing. We tried all kinds of ways to show whether these airborne microbes were active. So here we fed 13C labeled methane to one drum, 12C methane to another, and did stable isotope probing. This is all the work of Val and Kevin. So, if you've done stable isotope probing, you know that when the organism metabolizes the 13C labeled compound, its DNA is a little bit heavier than if it's just growing on regular compounds that have a very low percentage of 13C and are mainly 12C. And so those bands of DNA can be separated on a gradient and then captured and sequenced and amplified and sequenced to see, do we have an active organism that has actively taken up methane and incorporated that label. And so Kevin spent many happy hours running these experiments, running the PCR, wrestling with the data, and this paper was published in 2022, I think, and we did a lot of replications, and in a few of those, we see that there is incorporation of the label in the biomass. And so airborne methanotrophs can incorporate carbon from carbon-13 methane. And so the question is, okay, we know clouds are active. Clouds are definitely full of active microorganisms, but how about the rest of the atmosphere? How much remediation is really going on up there? So, in my career, I started with methane. I ended I'm ending with methane, continuing with the dioxins. But it's been a wonderful time. I just am so grateful to all the students and collaborators. Here's Kevin on a couple of trips. When he got to go to France for a year, me and Val got to go for a week, I think. And here he is with Anne-Marie Delort and Pierre Amato. They are the premier cloud microbiologists in the world. Here we are going by train to the top of a volcano to see their air and cloud sampling stations. And I'd like to, just show the funding that we got and my collaborators. And as I mentioned, the Tittabawassee working now with Gerben on that and folks at Michigan State. And finally, I'd like to, I don't think Maria's here, but acknowledge Maria Rivera, who's our building and laboratory manager, who's, she's not a member of my group, but she makes sure that our building and our lab runs well. And so that concludes my talk, and I'll just show a few other students whose work I didn't talk about today, but acknowledge their contributions as well. So thank you.
Into one of the microphones.
I had a question about the dioxin degradation. So the how my respiratory bacteria are anaerobic, right? So
Yes.
I was thinking, have you seen any sort of patterns of like if there's an area in of the sediment that gets like covered or area of the passage that gets covered in sediment by tides, whether there's like higher rates of degradation compared to like a different area that is like, just constantly like open to oxygenated water above.
Yeah, that's a really good question, and comes back to the key of how complex sediments are, right? That the water column is oxygenated and the upper few centimeters of the sediment are oxygenated through resuspension and the activity of little organisms that live there. And so you might think, and we believe it's true, that deeper in the sediments, you might see reductive dechlorination happening. And when you produce those dichloros or monochloro-dioxins, those indeed, if they make their way to that aerobic zone, might be completely aerobically oxidized. And that's what Gerben studies. So that coupling of an anaerobic and aerobic system is a really important key in remediation for a number of pollutants. But for sediments, that's a really keen observation because that is exactly what could happen. Whether we've measured that in situ, I think the answer is probably no, because it's too difficult, but certainly the organisms are there and they have that opportunity. And so it likely happens.
Yeah. Thank you for a wonderful, of course. The evolution question is always on my mind, and I brought it up a little bit at least talk. And of course, Max has something to say about this, but I always think about halogenators, right? They're kind of related to the sulfate producers or our elite sulfate producers. themselves. And those are specialists at keeping the electrons, at that low, very negative potential. Sorry to get a little technical here, but evolutionarily, how does that, where do you view that jump? You got the low electron, but then you're confronted with a very different compound. How do you make that? What was your view of that?
So the obligate OHR dehalogenators are not sulfate reducers. They're not. Some of the facultative ones can use various sulfur compounds as electron acceptors. Things like dihalococoides, it does not. So just from a purely theoretical standpoint, if you calculate the negative free energy for a dioxin and molecular hydrogen, that's yielding energy, right, at very low levels, incredibly low levels. It's crazy, but it is energetically feasible that they gain energy from that. And I think, and by the way, I think Max would have more than just a little bit to say about any of this, but I'll let him talk later. But I think when I first started, and Jim is an environmental engineer, I'm an engineer. When he first started enriching things, his early enrichments were out of the sewage treatment anaerobic digester. That's where strain 195 came from, an anaerobic digester in Ithaca, New York. All right, that's what engineers do. We want to study a microbe, let's go get sludge. So I think he might have thought, and others might have thought, these are new organisms. These are young organisms. They have evolved over the last not too long distance because we've just started putting all these compounds out in the world recently. But actually, that's not the prevailing hypothesis now. It's that in nature, you have this organohalide cycle, and there are naturally occurring halogenated organic compounds of, thousands or tens of thousands of different types that are out in the world in natural, pristine environments at very low levels. And that's why these organisms are out there. That's why they have 15 or 35 reductive dehalogenases, because they've evolved to scavenge these things out of the environment. So I know less about the ones that use sulfur, but they are also very interesting. So I can't really address that, but yeah. Thank you. Thank you.